Are you ready to uncover the hidden truths about our ancient past? What if everything you thought you knew about Neanderthals, those enigmatic early humans, was based on faulty evidence? Today, we're diving deep into a groundbreaking discovery that's shaking up the world of archaeology and rewriting the timeline of Neanderthal extinction in Europe, especially right here in Belgium. For decades, the narrative was pretty clear. Neanderthals roamed Europe, then modern humans arrived and, poof, they vanished. And for a long time, the spy cave in Belgium held a special place in this story, with its Neanderthal remains suggesting they clung on in northwest Europe much later than elsewhere. We're talking as recently as 37,000 years ago, that put their bones and tools among the most recent Neanderthals found on the continent. It was a really tantalizing thought, a bit like finding a remote village still clinging to old ways in a rapidly changing world. But here's the kicker, folks. When those radiocarbon dates came out for the spy Neanderthals, placing them so late, a few eyebrows started to rise. Was it too good to be true? Was there something off? Turns out those concerns were absolutely spot on. Scientists, being the meticulous lot they are, have been refining their techniques for radiocarbon dating. They've developed incredible ways to purify these incredibly old fossil samples, making sure that any pesky modern carbon material which could make the dates seem artificially younger is completely gone. Think of it like trying to clean a priceless antique. You need to be incredibly careful not to damage the original, but you also need to get rid of every speck of dirt that's accumulated over centuries. And guess what? This isn't the first time this has happened. Several sites that were once believed to be the very last strongholds of surviving Neanderthals were, after these new super advanced purification methods, redated to much, much earlier. It's almost as if the previous methods were giving them a bit of a longevity boost they didn't actually have. Now, in this groundbreaking new study published in a paper aptly titled Reevaluating the Timing of Neanderthal Disappearance in Northwest Europe, researchers went back to the drawing board with those Belgian Neanderthal remains. And what they found is truly astonishing. The overall picture that's emerging is far clearer. Almost all Neanderthals in Europe, it seems, went extinct roughly 40,000 years ago. This pushes back their final farewell in places like Belgium significantly, far, far earlier than previously thought. The secret weapon in this reevaluation, brand new cutting-edge chemistry methods. These techniques are the only way to truly decontaminate these crucial Neanderthal bones, making absolutely sure that every single contaminant is completely eradicated before dating. It's like forensics for ancient bones, leaving no stone unturned. One of the most striking examples of this contamination problem comes from a Neanderthal scapula. That's a shoulder blade for those not up on their anatomy, from the spy cave. This particular bone had previously given incredibly recent dates around 28,000 years ago. Imagine thinking Neanderthals were still kicking around that recently. But when the new analysis was performed, scientists made a jaw-dropping discovery. This bone was heavily contaminated with modern bovine DNA. Yes, you heard that right. Cow DNA. It turns out over 100 years ago, when these precious Neanderthal remains were first excavated and preserved, people used animal-based glues, likely made from cattle bones, to stabilize them. Back then, they had no idea that this seemingly innocuous preservation method would wreak havoc on future radiocarbon dating. It's a bit like trying to date a vintage car, but someone's painted it with modern car paint. You need to strip away all the new stuff to see the true original. So how did they fix this sticky situation? The scientists in this new study employed truly advanced radiocarbon dating techniques. They were able to date the Neanderthal remains by extracting a single isolated amino acid using something called liquid chromatography separation. Now, I know that sounds incredibly technical, but essentially, it's a super precise way to pull out only the ancient carbon from the bone itself, completely excluding any carbon from those pesky contaminants like the glue. This revolutionary approach allowed them to reliably date the bones, sidestepping all the issues that had plagued previous attempts. Those earlier dates, which were far, far too young because of the contaminants, can now be seen for what they were: misleading. In some cases, previous dates based on Neanderthal specimens from the Spy Cave were up to 10,000 years too young. That's a massive difference, enough to completely alter our understanding of their timeline. It's like finding out your great-great-grandparent was actually born a century earlier than you thought. And it wasn't just Spy Cave that got a thorough reevaluation. Researchers meticulously redated specimens from other Belgian sites too. The goal was to reduce the uncertainty around that crucial time window when Neanderthals finally disappeared from Northwest Europe. And the results are pretty consistent across the board. The majority of the new dates are significantly older than what we previously had for the same bone samples. We're talking up to 5,000 years older in some cases. Let's talk about some of the specific stars of this show: the Neanderthal skeletons discovered during excavations in Belgium, affectionately known as Spy 1 and Spy 2. Both were males, and their cranial volumes, basically the size of their brains, were around 13,500 cubic centimeters. To put that in perspective, that's well within the range of modern human brain size, perhaps even on the larger side. One particular specimen, Spy 94, an upper right molar, proved to be a goldmine of information. When directly radiocarbon dated using these new methods, it yielded an age range of 39,150 to 37,880 years ago. That's a pretty tight window, only 270 years, with a median age of 38,515 years, give or take 635 years. This molar is believed to have belonged to the cranium known as Spy 1, which was confirmed as a male through DNA analysis. But the plot thickens. Spy 94 isn't an isolated case. It's genetically similar to Goyet Q561, a female Neanderthal from the Goyet Caves, also in Belgium. And here's the exciting part: Goyet Q561 groups closely with other late European Neanderthals. Researchers managed to extract nuclear DNA from the right femur of this female Neanderthal, who lived approximately 43,000 to 42,080 years ago. This incredible discovery points to a continuity of the Neanderthal population in the region even before the massive eruption of the Campanian Ignimbrite, a supervolcano event that dramatically altered climates around 39,000 years ago. Now let's briefly turn our attention to another fascinating find, Engis 2. This refers to a portion of an assemblage discovered way back in 1829 by a Dutch physician and scientist, Philippe Charles Schmerling, in the lower Schmerling Caves. Engis 2 is essentially the partial remains of a Neanderthal infant aged around 2 to 3 years old, comprising a partially preserved cranium, fragments of an upper and lower jaw, a maxillary bone, and an upper incisor tooth. The Schmerling Caves are just north of the Belgian municipality, which is where this group gets its name. Just like Spy Cave, Engis 2 also had some dating controversies. There were 2 radiocarbon dates available for Engis 2. Scientists initially disregarded an earlier value of 26,820 years ago, plus or minus 340 radiocarbon years, because it was deemed too young, and you guessed it, probably contaminated. But the more accurate date using those refined techniques is 3460 radiocarbon years ago, which translates to a calibrated age of 34,590 to 36,110 years ago. Interestingly, the identification of Engis 2 as Homo neanderthalensis. Neanderthal and another specimen, Engis 1, as Homo sapiens, modern human, was mostly based on anatomical similarities and chronological parallels. Crucially, Engis 2 was discovered alongside Neanderthal Mousterian artifacts, a type of stone tool characteristic of Neanderthal culture. This association strongly supported its Neanderthal identity even before advanced genetic analysis. These groundbreaking findings really hammer home the importance of using robust pre-treatment methods when dating Paleolithic human remains. It's absolutely critical to reduce any contamination bias. Think of it like cooking. If your ingredients aren't perfectly clean, no matter how good your recipe, the final dish just won't be right. Scientists aren't stopping there, though. They're currently analyzing other archaeological evidence, such as bone tools, to further improve our understanding of the cultural transition between Neanderthals and Homo sapiens in this region. Were there exchanges of ideas? Did they learn from each other? These are the fascinating questions they're trying to answer. Now let's circle back to Goyet Q561, that fascinating female Neanderthal from the Goyet caves. We mentioned her briefly, but her genetic data is absolutely crucial. Researchers successfully extracted nuclear DNA from her right femur dating back an impressive 43,000 to 42,080 years ago. And the DNA analysis unequivocally revealed that Goyet Q561 was indeed female. What's more, as we touched on, Goyet Q561 is genetically similar to Spy94A from Spy Cave, and she groups closely with other late European Neanderthals. This isn't just a random connection. It speaks to the broader genetic landscape of Neanderthal populations in Europe during this critical period. These genetic studies from the Goyet Neanderthals have provided incredibly valuable data on the genetic makeup of these late Neanderthal populations. They've revealed typical Neanderthal genetic markers, contributing significantly to our overall understanding of their population structure. It's like getting a detailed family tree for an entire ancient species, and it gets even more detailed. Analysis of mitochondrial DNA, mtDNA, from the Goyet Neanderthals has shown something really interesting. They had distinct maternal lineages compared to other Neanderthal groups found across Europe and Asia. This suggests a certain level of genetic diversity among Neanderthals, which is a nuanced point often overlooked. We tend to think of them as one homogeneous group, but clearly there were regional variations. However, and this is a crucial however, while there was some regional diversity, the mitochondrial DNA of Goyet Neanderthals still falls within the broader framework of known Neanderthal maternal lineages, which overall are less diverse than those found in modern human populations. What does this lower diversity suggest? It hints that Neanderthals may have had smaller, more isolated population groups with less gene flow between them. Imagine small disconnected communities rather than a sprawling interconnected network. The Goyet mitochondrial DNA further reveals that these Neanderthals were part of a group with a shared maternal lineage that was present in Western Europe during the late Neanderthal period. This provides a clearer picture of their geographical distribution and genetic inheritance during their final stages. The findings from Goyet support a growing idea Neanderthals were far more capable and complex than they're often given credit for. The evidence suggests they were capable of complex behavior, including the sophisticated use of tools, not just crude implements, but tools showing real craftsmanship. They were skilled hunters mastering their environments. And intriguingly, there's even a suggestion of possibly symbolic acts similar to what we've seen at other European sites. This challenges the old stereotype of brutish cavemen. Perhaps one of the most exciting aspects of these new dates is where they place the Neanderthal fossils, incredibly close to the time when modern humans, our direct ancestors, were also present in Europe. This temporal overlap is absolutely crucial. It strongly supports the notion that interactions between Neanderthals and modern humans were not just possible, but potentially frequent. Imagine two distinct human species sharing the same landscapes, perhaps even crossing paths, trading knowledge, or maybe even something more. And this is where it gets truly fascinating. Genetic data from these Neanderthals contributes significantly to understanding the gene flow between the two species. It helps us piece together how Neanderthal genetic material was integrated into the early modern human gene pool. That's right, many of us walking around today carry a little bit of Neanderthal in our DNA. It's a tangible link to our ancient past. The Neanderthals from the Goyet caves represent an absolutely essential piece of the puzzle of late Neanderthal populations in Europe. The combination of fossil evidence and genetic insights from this single site provides an unparalleled window into their lifestyle, their genetic diversity, and those crucial potential interactions with early modern humans. These findings enhance our understanding of Neanderthal behavior in so many ways. Their sophisticated tool used their social structures were they tribal family units, and their remarkable adaptability during the incredibly challenging climates of the Ice Age. Overall, the Goyet Caves contribute profoundly to the narrative of Neanderthal existence, highlighting their complexity as a species and their undeniable place in human evolutionary history. They weren't just a dead end. They were a vibrant, adaptable part of our story. Now let's shift gears slightly and talk about another fascinating individual from the Belgian caves, Goyet Q1161. This isn't a Neanderthal, but an Upper Paleolithic individual, meaning a modern human. Genetic analyses of Goyet Q1161 have given us some incredible insights into physical characteristics like pigmentation, eye color, hair color, and even genetic lineage. It's like getting a glimpse into the appearance of our very early European ancestors. Here's what we've learned. The genetic profile of Goyet Q1161 suggests this person likely had relatively light skin, however not as light as many modern Europeans today. Early European hunter-gatherers often showed skin pigmentation adapted for regions with less sunlight exposure, but complete depigmentation probably wasn't present at this very early stage. Think of a slightly warmer, richer skin tone than what you might imagine for someone living in a cold climate. As for eye color, genetic data from similar individuals in the Upper Paleolithic, like other European hunter-gatherer groups, suggests a range that could include dark brown eyes. While blue or lighter eyes became more common in later populations, at this point in time, dark eyes were likely prevalent. And hair, Goyet Q1161 probably had dark hair. Early European hunter-gatherers, including those with the same genetic background as Goyet Q1161, tended to have dark hair as the dominant trait. These characteristics are super important because they indicate that Goyet Q1161 and related populations were well adapted to the European environment at the time. And crucially, they contributed to the rich tapestry of genetic diversity we see in later European populations. It's like seeing the foundational colors being laid down for a magnificent painting. The Goyet Q1161 fossil is believed to be around 35,000 years old, placing it firmly in the Upper Paleolithic period. This era corresponds to the Aurignacian culture, which is one of Europe's oldest known modern human societies. These Western hunter-gatherers, like the individual from Goyet Cave, were among Europe's earliest inhabitants during this period. Genetic analyses have confirmed that the Goyet caveman, as he's sometimes informally called, was a male. Goyet Q1161 was identified from human cranial remains discovered in Belgium's Goyet Caves. The analysis of this specimen primarily focused on the skull, which provided incredibly valuable genetic and morphological information for understanding early European populations. The Goyet Caves are truly a treasure trove of human history. This fossil is part of a collection of both modern human and Neanderthal remains from the site, which has provided immensely important insights into the life, culture, and genetics of early modern humans in Europe during the late Pleistocene epoch. And here's where it gets mind-blowing. The Neanderthals and the Goyet Caves were among the very last of their kind surviving in Europe. They existed during a time of intense environmental and competitive pressure, a challenging period indeed. But the truly astonishing part is the Goyet caveman, Goyet Q1161, who, as we mentioned, had a combination of modern human and Neanderthal DNA. This exemplifies the early exchange of genetic and cultural traits that would go on to shape future European populations. It's a testament to the dynamic interactions that occurred between these two distinct human groups. And for those of you wondering, why does this matter to us today? Well, Neanderthal genes in modern humans actually influence a variety of traits, including our immune system responses and even adaptations related to skin. So a bit of ancient history is literally running through your veins. Understanding precisely how and when Neanderthals vanished has been one of the hottest, most debated subjects in paleoanthropology. These new findings from Belgium contribute significantly to resolving some of those long-standing questions. While the overall pattern indicates most Neanderthals went extinct around 40,000 years ago, it's fascinating to note that there were still a few small resilient groups like those at Spy Cave that did manage to survive for a few generations after that 40,000-year window. They were clinging on perhaps in isolated pockets for a little while longer, and with that tantalizing statement, the idea that a few tough Neanderthals might have held on just a little longer, we leave you to ponder the mysteries of our shared human history. It's a story of survival, adaptation, interaction, and ultimately a vanishing act that continues to captivate us. Thank you for joining us on this incredible journey through time. If you found this as fascinating as we did, please hit that subscribe button, give us a thumbs up, and share this video with anyone who loves a good historical mystery. And don't forget to explore our channel's other highly compelling videos. Until next time, stay curious and stay questioning. Take care.